For today's webinar, we are very, very excited to have Dr. Mohammed Sarayan give us a talk on robust cancer mutation detection with deep learning models derived from tumor-normal sequencing data. Dr. Sarayan is currently a senior principal bioinformatics scientist at Roche Sequencing Solutions, working in genomic data analysis. And before joining Roche, he was a postdoctoral researcher at UC Berkeley. 2013, he earned his PhD in electrical and computer engineering at Texas A&M. He is co-author of omatic, the first convolutional neural network-based approach for cancer mutation detection. And based on this AI variant calling approach, he led Roche's participation in the Precision FDA's Truth Challenge version 2 with the best performer recognition in 2 categories. He is particularly interested in developing new solutions to genomics problems that leverage recent developments in deep learning. Dr. Sarayan, we're pleased to have you here. Please share with us your findings.
Thank you very much for the nice introduction. And I'm very happy to present my work here. Let me share my screen. Okay. Hello, everybody. So today's talk will be about how we can use deep learning in cancer mutation detection using the tumor-normal sequencing data in the SEQC2 consortium. So first of all, just very quick introduction about somatic mutations in cancer. You know that during normal organismal aging, cells are exposed to DNA damaging events that eventually results in cells harboring some mutations which are not inherited. So like this red mutation in one of the cells. So these mutations occur sometimes in the regions of the genome that lead to uncontrolled cell proliferation. So this ultimately will cause tumorigenesis and rapid accumulation of more mutations. And such mutated cells may later metastasize and invade other parts of the body. So the important signature of these mutations is that they are not inherited genetic alterations. So basically, which means that in the normal cells, you should not see them, but in the tumor cells, you will see them. And these are very critical signatures in cancer because they drive the cancer and tumor genesis. And for detecting them, one approach would be to compare tumor and normal samples because, as I said, they are not in the normal, they're only in the tumor samples. But there are some challenges in detecting somatic mutations. One of them is contamination, which means that it's not that when you sequence the normal sample, it's always pure. So there may be contaminated with the tumor cells. So if you sequence the tumor and sequence the normal, you may see some somatic mutations in normal. So if you just subtract the mutations between tumor and normal, you may lose some of the real mutations. So it's not always perfect. Another problem is tumor heterogeneity, which means that there are multiple populations of cancer cells in the tumor. This happens as follows. For example, first the founder cell will form, which is shown as a red cell with a red mutation here. And then this cell divides and we'll have more copy of this cell. And at some point, one of these red cells may acquire another mutation like the purple one here. And that can generate a second population, and this can continue. So at the end, we will have multiple populations in the tumor. And when you sequence them, each mutation will have its own allele frequency, like the fraction of reads that support that mutation. So we should be careful about that because these multiple populations of the cells will have different allele frequencies. So as I said, they are tumor specific, which means that they are ideally not in normal, they are in the tumor. So if we sequence the normal samples and sequence the tumor samples, and then we compare the sequence with the reference, if you observe mutations which are in both tumor and normal with high frequency like this one, then and this is not in the reference. So this is more likely to be a germline variant because it's happening both in normal and a tumor. But if it's something which is only in the tumor, that's more likely to be in the tumor. This is more likely to be a somatic mutation. So the current approaches to identify somatic mutations are usually based on statistical modeling, which means that their goal is to identify mutations which are less likely to be in the normal, like tumor mutations which are less likely to be in the normal. Some example algorithms are Mutate2, Stralka, and Derived, and they are actually relatively good for design condition or sample types, but usually they're difficult to generalize to different conditions. One machine learning approach that often have been there was somatic-seq, which uses machine learning to integrate different methods. It's not the standalone method, it's based on ensembling the different colors, and basically it used the output of different colors as the starting point to basically identify some features from each of those mutations from different colors and train a machine learning model on top of those outputs to decide whether these are real mutations or not. But one problem is it's limited to just output from the other algorithms, and also it cannot fully capture raw information because it only, basically the machine learning model is trained on some manually extracted features rather than looking at the sequencing data directly. So we know that convolutional neural networks have significantly improved classification tasks. For example, in image classification, object detection, segmentation, and natural language processing. And this has been very helpful and even actually on biology, like biological image analysis or gene expression analysis or epigenetics, single cells, and even germline variant calling. And these deep learning models have been used. For variant calling, you probably have heard of deep variant, which is the first CNN-based germline variant calling approach. So as I said, this is a classification task because, for example, for germline variant calling, you want to classify whether an observed candidate variant is true or what's the basically genotype of that variant. Is it a reference or is it homozygous or heterozygous? This is how the variant works. So it generates like a pileup image of the reads, something like what you see in some programs like IGV, for example, visually. It generates something like that, and it gives it to a deep learning model that will classify the variant. One issue with the variant is that it needs, first of all, a very huge network to work because the data is kind of big, like the size of the image is, because it looks at all of the reads aligned to the genome. It needs like a large network to be trained on. But what we did for neosomatic, which is our approach for somatic mutation detection, and actually the variant currently doesn't work for cancer, it's only for germline. But what we proposed for neosomatic, which is our approach for somatic mutation detection, is based on a smaller neural network. I will explain it. And this is published like 2 years ago in Nature Communications. So the way that we actually model the data, we try to capture the main genomics context information in the input channels of the data that we give to the network. And we use a very efficient data prep, training, and testing environment, and the model. It can be easily trained and on different like sequencing model or error profiles or different sample purities. And also it can be used as a standalone or ensemble approach. So I will explain the details of these in later slides. So it can efficiently learn error models to identify the real mutations. This is like one example input image that we give to the network. So first of all, what we do to classify somatic mutations, we first scan the genome, which means that we just check the pileup. This is like not the deep learning part. This is just scanning the genome, identify candidate mutations, which are mutations in the tumor which have enough difference to basically enough allele frequency or enough read support, and they are different from the reference. So basically, we'll have a threshold for allele frequency cutoff for allele frequency, and that will be that will give us some candidate mutation. So usually, like 99% of those are just artifacts if the allele frequency is small, or they are germline variants. So the task of the network is to basically say whether this is a somatic mutation and what is the type of the somatic mutation. Is it SMV, insertion, or deletion? So for each of those candidate positions or candidate mutations, we generate, first of all, we generate 3 matrices. These are formed basically by looking at the alignment of tumor reads and normal reads to the reference. Let's say our candidate location is a CNV C to T here at this middle position. So we have identified that based on like just checking like the pileup, read pileup at this position, and we see that okay, there are some reads that are different from the reference. Now to form the input matrices, we generate a augmented alignment of the tumor and normal reads to the reference, which means that we look at the bound file or the alignment file, and wherever there are some insertions in the read, we add gaps in the reference, such that at the end, this looks like a multiple sequence alignment of tumor and normal reads to the reference. Although we will not do the multiple sequence alignment, we will just, each read is aligned pairwise to the reference using, for example, BWA aligner. But to generate the final matrices, we form this, like visually, we form this multiple sequence alignment like image. And then based on this alignment, we generate these matrices. The reference matrix with measures in each column of this alignment, what is the base in the reference. For example, here we have TGG. So this matrix has like 5 rows for gap and ACGT. Sorry. And the columns are like the number of columns in this alignment. For example, here it's like 18. So in each column, it just checkbox the reference. So it's like T, G, 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 and in the middle we had the C. So it's like 0, 1 matrices based on the reference sequence. And for the tumor matrix, tumor count matrix, it's similar, but instead of 0, 1, we put the number of bases for each of like ACGT or gap. For example, here all of them are T, so we put 6 here. Or like in the middle, we have 3 Cs and 3 Ts, so we have 3 here and 3 here. So like that, we form a tumor matrix, and similarly, we form a normal matrix, normal count matrix. So these 3 matrices are the initial channels, input channels of the which goes to the network. It's like for natural images, you will have RGB channels. Instead of RGB, for example, we may have reference, tumor, and normal count matrices. And actually, if you visually compare these two, you may identify that okay, there's something happening here because this column is different in the normal and tumor, and they're different from the reference. So basically, network will also learn these features directly from this raw data. This is another example for deletion. Everything is the same, only we can see that when you have a deletion, you will have some gaps in the middle. So when your counts will have like more numbers in the first row for the tumor matrix. And insertion similarly. So for insertions, we will have gaps in the reference. We will add them because of the augmentation. And we will have the bases in the tumor, but in the normal, we don't have them. So you will have some numbers in the first row of normal, but most of the, there may be some, let's say if it's a heterozygous one, there may be some large numbers also here, but you will also have some bases aligned here because of the insertions. And the reference is all zero, all gaps. So these are the three input matrices. And then, so this is the reference tumor and normal. And then we also have more additional channels like coverage, information, position, which means that which column in the alignment is the candidate location, and additional features like base quality, mapping quality, strand information, and this kind of thing. So they try to capture like for each position and each base, what is the average base quality or mapping quality or whatever. Up to this channel, this will give us the standalone version, which is, it works by itself. It doesn't need any other algorithm to be used for. But we may also add some additional channels from other colors. For example, we may have a channel that says whether Mutate2 has called this candidate location as a variant or not, or what is the quality score for that, this kind of thing. So we may add any additional channels from other colors to support this variant or to not support this variant, this kind of things. And this will generate like the channels and like the input channels for any candidate mutation. So as I said, if you imagine like the image classification, in image classification, you have 3 channels for RGB. Here we have more channels for each candidate. And this goes to the network, which is a quite simple network. It has like 9 convolutional layers designed for this kind of input matrix that we have. And it has like some ResNet-like blocks with like some shortcuts. And on the top, we have a 2 classifiers, one for the type of the mutation, whether it is a SMV, deletion, insertion, or artifact, which is a non-class. And the length classifier that measures, for example, for indels, the size of the deletion and insertion, and position, which try to make sure that the classifications are for the target location that we had, target column that we had. So the workflow will be, we scan the genome to find the candidates, and then we prepare some input matrices and annotate them for those candidates, like for each candidate. By annotation, I mean, this is for training. So by annotation, I mean, we will know what is the class, each class, basically, what's the type of the variant and what's the length. Those like annotations should be added for each input. And then we train the network. And for testing, it's similar. We scan the genome and we prepare those input matrices for each candidate location. And we test the train model from the training step. And there are some post-processing to generate the final VCF, because the output, like for example, for indels, we may not know exact lengths because we have 4 classes for length classifier, which is 0, 1, 2, and more than 2. So if the length is more than 2, we identify the exact length in the post-processing to output the final variant, like the final variant in the VCF format. And this is like the computational complexity. This is based, I mean, the numbers is based on like the earlier work. So now it's a little bit like actually more efficient. But let's say this is like what we have in the current release. So for whole genome 30x data, scanning the alignment like in a 28-thread machine with this like a specification, it's like a 2-hour scanning the alignment to identify the candidates, 4.5 hours for input preparation, 45 minutes for predicting the network output, and 3 minutes for post-processing. And if we want to run in the ensemble mode, we have like 6 hours to run other colors. For whole exome, it's much more efficient, like I say, 120x, 3 minutes for scanning alignment, 2 minutes for input preparation, 1 minute for network calling, and 1 minute post-processing, and 30 minutes if you want to call others or colors. And for training, for example, using 8 Tesla V100 NVIDIA GPUs with 32 gigabit of RAM and batch size of 80,000, Training time actually depends on what is the training data, but let's say we have a one sample training, for example. In each epoch, we will have like 10 million candidate locations. This can take around like 270 seconds, like around 4 minutes. And like if usually the training will be done on in 600 epochs, so it can be finished in 48 hours. But again, it depends on how many samples you use for training. It can be more. It's just one sample, for example, or two samples. So to benchmark this work, I mean, in our original paper, which was published in Nature Communications, we use some synthetic data sets, which are based on Dream Challenge data set. They use the algorithm called BAM Surgeon to generate the synthetic tumor data. And the way that it works, it starts with a normal alignment and select some reads, basically randomly select some positions first, and on those positions, randomly select some reads, and it collects those and their pair and mates. And basically it injects the mutations or like alters the bases in the read such that it supports the like that random mutations that we want to add. And then they align back the those mutated, synthetically mutated reads to the original alignment. So what we will have is a BAM file or alignment file that has these mutations in them. So it's kind of tumor mutations in this sample. So if you compare these two BAM files, you should identify these mutations. So we tried this with different priorities and compare 6 other colors and also somatic sec, which is the ensemble color. And for each data set, we will have one trained model for different purities. And to evaluate, basically, we train on half of the genome and we test on the other half of the genome. It's like randomly selecting the regions in the genome. This is like the general accuracies based on F1 score. So we have new somatic, which is the ensemble new somatic, and new somatic S, which is the standalone version, and some other color, somatic 6, which is again ensemble color, machine learning based ensemble color, and these are like standalone colors. You can see for SMVs and indels, we are for both of them, we are more accurate than other colors, and the gap can increase like up to like 35% F1 score for like lower purities. And basically for more challenging situations like lower purity in those, this gap is more. And we will have a gap actually, like we will have like improvement over both somatic 6, which is the ensemble color, and also on the comparing to other individual colors, we have better performance. Another like data set that we use, which is again synthetic, is based on mixing 2 normal samples. For example, we can use some genome in a bottle or platinum samples, which we know the ground truth germline variance for them. So if we mix them, we can generate some kind of synthetic tumor sample for which we know that actually the somatic mutation or synthetic somatic mutation. So we can use that for both training and evaluation. And on this data set also, we observe significant improvement to other algorithms up to 15% for ensemble or 7% for standalone. And this is like to show how the coverage impact the performance. So here we have like different coverages of the data, 20x to 100x. And for SMVs and indels, we observed that the robustness of like neosomatic is much higher than others. So some of them fail much faster when you go to lower coverages, but it can preserve its accuracies to lower coverages compared to others. And this is also interesting observations. If we, in this example, actually, we compared the F1 score between the pure, when we have the pure normal, or when you have like 5% contamination in the normal, like 95% pure pure normal, which means that 5% of the reads will have some somatic mutations in there. And these numbers shows like how much drop in F1 score we will have if we go from 100% to 95%. You can see for neosomatic, both of their versions are quite robust and the drop is negligible, but other algorithms usually have high drop, like for mutate, it can be very high. Like best standalone alternative is Astralca, which is still not that bad. So far it was like synthetic data that we used for evaluations, but recently the SEQC2 consortium generates the very like useful, comprehensive and well-characterized somatic reference samples, which are like a tumor-normal match samples for a breast cancer cell line data. And they characterize the somatic mutations in there. It's like around 40,000 validated somatic mutations in whole genome. And you can see like the distribution, like the allele frequency distribution of those variants. They are from very low AF to high AF and around 37,000 SMVs and like 1,500 in those. These are like validated variants. So we will be sure that they are there. And also this is comprehensive, which means that anything else that you identify in the high confidence region of this data set should be false positive. So we can basically be sure about both false positives and false negatives, which is very useful for both training and evaluating this. And this is kind of the first data set that has this number of mutations in that. And another like advantage is it has like many replicates, like 123 replicates of these samples, sequenced with different conditions, different like whole genome, whole exome, targeted ampli-seq, from 10x to like 200x data coverage, with different platforms and different sequencing centers like HiSeq, NovaSeq, and different centers, and different titration to form different purities like 5%, 100% tumor purities, and again 10x to 300x coverage. And it has both FFPE and fresh data and the different library preparation. So it's very comprehensive set of samples that will be used or that can be used to evaluate the performance in different conditions. So our goal was to explore different model building strategies on this data set. So here I compare like 4 main models trained on neosomatic with this data set. The first one is based on the Dream challenge, so it's independent of this data set actually. This is just to compare how we perform if we train on a different data set, which is like Dream data set, and test on this like SEQC data set. So for that data set, we had, like, as I said, in silico spiking mutations. It's like 450,000 somatic mutations spiked in those samples, and 29 million candidates. So most of them, as you see, are artifacts or germline mutations. So basically, these are like 28.5 million of those candidates will be artifact, but the others are real somatic mutations. The second model we build is based on, we pick like 10 whole genome normal replicates of this SEQC2 dataset and generate synthetic tumors by spiking mutations. This is like, the reason that we did this instead of using the real tumor that we have here is we can have much more mutations, like 2 million somatic mutation and 27 million candidate locations on these 10 samples. So we mix the training data from the 10 samples in the training process. Then we use this model, which is called GT, ground truth 50, which means that we train on 50% of the ground truth sample, like the ground truth cancer sample. So here, this time, the real mutation are coming from the real cancer, so they are really not the synthetic, but real mutations. It has a lower number of mutations compared to the synthetic one, but still quite good number when you have 8 samples is like 400,000 somatic, and the number of candidates, you can see 137 million. And finally, we have a model trained on mixture of these two data sets to take advantage of both higher number of mutations and also some real cancer data, which is used in this one. We also have some alternative models in the paper. So there will be, actually there is a bioarchive paper for this part of the work, and it's actually submitted, it's under review, but you can actually see this data on the paper as well. So yeah, we have some additional models for whole exome, FFPE, or mixtures, and some sample-specific models. So this is like the overview of how we perform, or how different algorithms perform on this data set. So in this image, we have 123 columns, which are for 123 replicates that we have. So these are different like type of the replicates, like whole genome, titration, library preparation, whole exome, FFPE whole genome, and FFPE whole exome, and ampli-seq. So we have in total 123 columns. And in the rows, we have like 4 different model trained on new somatic, 4 different model trained on new somatic standalone, and other colors. And the color basically show for each, basically the way that we form this image, for each column, we identify the best algorithm among all of these methods. And then we compute the F1 score for that. And then we subtract the F1 score of other algorithms, or all of the algorithms, from that best F1 score. So if the number here is 0, it means that you are the best. Like basically, if it's lighter, it means that you are the best, or you are closest to the best performance for that replicate, because each replicate has its own limitations and has like the maximum. For example, if the coverage is very low, you are not expecting to get as good F1 score as when you have very high coverage. But what we want to show here is, in different conditions, which algorithm stays on the top compared to others. So if you are the lightest color, which means that you are the best for that specific replicate. And as you see, on average, these new somatic models are having lighter, basically, colors compared to others, which means that they are more robust or more consistent, consistently the best approach across different conditions. And also in terms of mean and median F1 score, if you compare like the best F1 score that we get to the best alternative standalone or like individual color, you can see we have around 6% mean F1 score improvement for SMVs here. And this is the same plot for indels. And this time you can see around 9% improvement compared to the best alternative, which is Lancet in this sense. So again, we can see it's a little bit less consistent, but it's still much better than the other algorithms, because indels are usually more challenging. This is the accuracies or F1 scores for whole genome samples or whole genome replicates, which you can see the reds are the new somatic. If you compare with the other colors, you can see that they're always ahead on average in terms of the F1 score. Now, this is a similar plot on whole exome data. Again, you can observe that the new somatic based models perform quite good. And this is actually, I want to show the titration example, which shows different coverages and different purities. So each column has a different coverage for the Tumor-Normal, and the size of the circle shows the purity from 5% to 100%. And again, comparing F1 score for different algorithms, you can see for different coverages and different purities, usually the F1 score is higher for new somatics models. And similar to the robustness to normal contamination plot we had before. So if we change the 100% normal to 95% normal, what will happen? That's what we want to show here. So in the x-axis, we have F1 score for non-contaminated normal case, which means that 100% pure normal. And the y-axis is F1 score for 5% contaminated normal, which means that 95% pure normal. So ideally, you want to be on the diagonal, which means that your performance is consistent. And also you want to be on the top right because you need higher F1 score, which is the case here for new somatic. You see the red dots usually are very near, very close to the diagonal. But some other algorithms, for example, Mutate2, Lancet, and Muse have a large drop. So it's like 40%, 50% drop of F1 score. Some algorithms like Strelka is also pretty robust. It's maybe not as good, but quite good in terms of the robustness to this contamination. This is also somatic sniper, which is robust, but its F1 score is not that high. Also for different insertion deletion sizes, here we have on the x-axis the size of the indel. So negative numbers are deletions and positives are insertions. And the F1 score you can see for different algorithms. Again, large improvement is observed, like up to 24% for insertions and 7% deletion improvement for more than 2 bases, the indels, which are the challenging cases. And allele frequency ranges, like different allele frequency, like 0 to 5%, up to 50 to 100%. You can see again consistent superiority, and also for more challenging cases, it's the advantage is higher. And another actually interesting observation is the performance on difficult genomic regions. So you know some regions of the genome are difficult for Illumina data, like for example, if they have tandem repeats or if they have segmental duplications. For tandem repeats, you may have like more artifacts in the reads, and also the alignment is not perfect because of the complexity or low complexity of the genome positions in that location. So the reads cannot align well, and they may have more errors in them. For segmental duplications, because there are like very homologous regions of the genome with that context, so reads will not uniquely align to those regions. So they will have very low mapping quality, and they might align actually wrongly to those locations. So these will generate artifacts, both false positives and false negatives. And for short reads, because they cannot expand outside these regions, it's more difficult for the Illumina data to align. Here we try to show how much performance drop you will have if you go from whole genome analysis to only the difficult region analysis. So the dark color, dark blue, is when you have the performance on the F1 score performance on the whole genome. And the lighter is when you only restrict to the difficult regions of the genome. You can see usually for new somatic, the performance doesn't change that much. In some cases it does, but relative to the others, it's much more robust. Usually, for example, for whole genome or even for titrations. And in general, SMVs actually are more consistent. But even for SMVs, when you compare other algorithms, you see large drop, maybe like 20% drop when you focus on the difficult genomic regions, which is quite significant drop. But new somatic is more robust to those region changes. And another observation is about FFPE. If the samples are FFPE, so they have some artifacts from FFPE process, but you can see for different times in FFPE, and when you pair it with normal or with fresh or FFPE normals, these are still outperform the other colors usually in most cases. And for library preparation also, if we have different DNA amounts and different protocols of library preparation, still we can see superior performance with neosomatic models. And we should be careful that all of these data, these are like these different library preparations or different FFPE or different, let's say, whole exome. These are not used for training. We only use 10 or 8 whole genome samples for training, which are normal, like whole genome samples. So this is like kind of out of distribution performance. So this was like the comparisons on the SEQC data. And in terms of like how scalable neosomatic is, in our paper, we had a collaboration with Microsoft to use Microsoft Azure to run 261 whole exome cancer samples from TCGA in 3 cancer types on H16 instances, which have 16 cores with 112 gigabytes of RAM. And we evaluated the accuracies on like 45,000 almost experimentally validated SNPs. So we want to both show like how the runtime works and how the sensitivity is. And you see per sample, like for these like 260 samples per sample, the runtime for standalone is less than an hour, like 0.7 hour. And for the ensemble, it's like 2.4. It's like the most efficient in terms of the cost way of running this. Again, this is like based on the original paper. Now it's much more efficient, but this is just getting some sense how fast or how expensive it can be. And it can, you see that most of them are less than $1 per sample for whole exome. And it can give you, for example, for ensembles up to 99% sensitivity. So in summary, SEQC2 train model, both using spiking mutations and the ground truth. Somatic mutations boosted the somatic mutation detection performance. And neosomatic shows that it significantly improved in challenging cases in those low purity, low coverage, high sequencing error, or difficult regions of the genome. And it was very robust to different kind of conditions like coverage, FFPE, process, Tumor-Normal purity variations. And it can be applicable to different sequencing strategies. Like, for example, for the main paper, we actually even compared with PacBio data, and it shows very good robustness. And source code of this work is publicly available on GitHub in this address. With that, actually, I should acknowledge my collaborators in Roche sequencing, my colleagues, Margoub, Hugo, Litai, and Roger. And on the FDA side, Wenming helped a lot with this SEQC work. Thanks, and I will be happy to answer any questions.
Mohammed, thank you very much for that overview of neosomatic. That was definitely a tour de force and very impressive results. We do have several questions. So one of them you've already answered. Gujan Hariani asked about, is it open source? And you just gave the link for the code. One question is, do you think it can work beyond tumor-normal pairs? I think most of the data that you had were in the context of tumor-normal. Have you examined neosomatic in the context of tumor only, for example?
Yeah, that's a good question. Actually, yeah, tumor-normal is what we have originally worked on the paper and also in this SEQC work. But we have started looking on tumor only, and internally we have analyzed that and shows good performance, but it's not ready for release yet, but hopefully very soon we will have that version out. Okay.
And she has some more questions. Let me get to some other people first, and I'll get back to some of the remainder of hers. So Dr. Webgen asked, does your approach work for, say, in liquid biopsy context where you're using UMIs to do corrections? Have you looked at this in the context of, say, UMI-based data? And do you feel like you would continue to see this improved performance, even at lower allele frequencies? I know you had a slide, showing the different performance at low allele frequencies, but if you had UMI corrected data, do you feel like that would improve things? And how would that be incorporated? In other words, would you give the UMI corrected data, or will you actually give, all the data to new somatic and let it do the correction?
I think we have explored a little bit in that context, but there's more work for definitive answer. But I guess in general, maybe just directly using somatic or with this structure, it's better to first filter or correct the data using UMI correction. Mm-hmm. But if you have another maybe network to do that step, also deep learning based, it may improve. But you may actually change somatic to somehow identify these clusters of reads that have the same UMI somehow. But with this current setting, I think probably UMI corrected is the best way to do this. And I expect, as I said, I didn't explore a lot yet. We are working on those kind of samples, but I guess because even with UMI correction, you may still see some noise or some level of noise, so it can still improve, I guess. Yeah.
Okay. And so your colleague Wen Ming has a question, and I think it's just more to maybe express what is generalizable from the data sets that you trained on versus maybe other contexts. But he asks, do you feel like a lot of the data was derived from, for example, in your working group, you worked with a triple negative breast cancer cell line, tumor-normal. And do you think the network, the convolutional neural network that that you got out of this, that was trained based on this data, how do you think it generalizes to other cancer types, that may have very different mutation characteristics in one sense? I mean, different, key drivers or things like that. But, or I guess maybe a more general question is, what would be the conditions that you would think you would have to retrain, when you get into a certain oncology context?
Yeah, I think this is kind of difficult to answer, but one thing that, for example, we can observe even on this data set, you can see the Dream Challenge model. This is trained on Dream data, is completely blind to this SCQC data. It's not as good as when you train on SCQC, but it's still better than other standalone colors like Astralcar or whatever, which means that it's to some extent robust to this. It's not trained only on this sample. And also we have tested that on different conditions here, different purities, different. So I don't think cancer type will be very, very critical feature here. More important feature, which may actually change the performance, is sequencing error profile. For example, if it's a different sequencer with different errors, which is not observed or was not basically trained on this data set, it may cause some deviations. But I expect that in general, if it's the same aligner and same sequencing technology, it should usually perform pretty good. And actually, we made this model public in the repository, so and we use it as a default model. But again, to get the best performance, if you have enough training data, you may retrain or continue training starting from this model, or retrain on a few samples that you have to make sure that error profiles are still the same, something like that.
Yeah, and I suppose one of those challenges there is having a good training data set. Certainly finding truth is not always easy, especially in somatic situations, right? Because they can be of low frequency, and of course they're often novel. And so, determining truth to get a large enough set. In other words, you can determine truth for, maybe a few hundred rather easily, even with PCR methods or other methods, but getting truth in general on such a wide set, is sometimes a challenge. Okay,
yeah,
go ahead.
Actually, what we usually do is to use, if you have multiple replicates of normals, you can use that BAM surgeon approach that I mentioned to generate synthetic. Actually, the third model we have here is based on that, or even the Dream Challenge model is based on that. So here we didn't use the real tumor data. We use the spike in mutations, and you see the performance is not that bad. It's very close to when you use the gram truth. So
yeah, that's actually very impressive and useful in terms of looking at other contexts. Yeah.
Yeah.
All right, so we have a question from Jim Willie. He asked, what do you think are the primary characteristics of new somatic that allows for its improved performance? In other words, is it the actual deep learning part, the convolutional neural network? Is it some feature set that you, the way you framed it in terms of how you originally defined the input channels, or what do you attribute the improved performance?
I think it's kind of combination of all of these, but mainly I guess the deep learning part, because usually people, the way that try to classify these mutations, basically the main problem is to classify the noise from the real mutations. And the way that it has been done so far is trying to statistically find some features that, for example, if some feature is larger than some threshold, it's real or not. But these thresholds are kind of arbitrary. But deep learning can just learn this from real data itself, and it can see more stuff that even visually or like a human cannot understand. So it can really see all of those features from the raw data and learn that with different conditions when this is a real or when it is not real. Additionally, and actually, this is in general about deep learning, but one important thing is how you give the data to the deep learning network. So we try to make it very easy for that to understand. For example, this reference augmentation that we did, we basically make the insertions very obvious in the alignment. For example, deep variant or other deep learning variant columns, like germline variant colors, originally didn't have any basically the insertions were just positions of the insertions, not actual sequences inserted. So I guess this new approach that we have will make it more clear for the network to decide. And also because it's simpler, simple network structure, it can easily be trained on very huge data sets like this one, which is as I said, 200 million candidates, something like that, which can be very huge. But in general, the main advantage, I guess, is coming from deep learning how to basically identify these artifacts from real mutations in different conditions from the real data. Okay.
And I had a question of my own before we get back to maybe some of the secondary questions that individuals had. So, you talked about in your presentation the impact of tumor purity, and you also mentioned, potential impacts of tumor heterogeneity. Is there an output, or do you think there's part of new somatic is some output that tells you information about the purity of the tumor, just because that may be useful by itself to know? In other words, if you showed that how in general, just for all callers, performance will degrade with lower tumor purity, but I didn't know if you had an output from it that would give you information about tumor purity as well as maybe heterogeneity, or do you know, is that something that the new somatic also learns, or do you think it's possible to glean that information from the implementation of the model? Just how do you, what information is available in that area?
I think the current architecture cannot handle that because it's like learning per position or in a small neighborhood per position. To measure the purity, you need to have a big picture across the genome, how the allele frequencies are distributed. I don't think this model will do that, but there may be some changing the input, the way you input the data to the network or different network structure. Probably that's a good research project, but I don't think new somatic by itself can learn that, because as I said, it just see one image from one candidate location. It should see different regions of the genome to say what's the purity. Gotcha.
Okay. Well, let me go back to some secondary questions that some of the same people had. So Gunjan also asked about whether new somatic, trained, let's say, in whatever context that may be more whole genome, or let's say, in some synthetic whole genome, whole exome, if you could apply that to a specialized panel, like to a panel that was a mega base or less, or if you need to do some special training for the panel just because panels sometimes maybe have their own characteristics. Any views on whether you can immediately take this to a small panel, it would perform as well, or do you think that that would be a situation where you need to do specialized training?
I think we have tried some of those, and actually here we have like implicit data, which is kind of a small panel. I mean, it's not exactly the same, but quite similar. And it performs well. One difference is the coverage. When you have a small panel, usually the coverage is very high, which may be different from whole genome training. I think it should work relatively good, but if you can generate, as I said, synthetic tumors on those samples and retrain or continue this training, that should get more advantage performance, I guess.
Okay, and then maybe the last question, just due to time. So Jim Willey also asked that, and I was surprised as well, that the performance of all the callers, or at least most of them anyway, didn't seem to be, noticeably or horribly worse with the FFP samples. Was that surprising to you, or do you think that may be due to maybe the FFP versions of the samples aren't really as bad as real life, FFP samples?
Yeah, I think the allele frequency cutoff that we have for this ground truth data may be higher than the level of artifacts that the FFP samples here had. But yeah, if you go very low AF, it may be worse. Yeah.
Yeah, and do you think that it may be possible to use that kind of framework to learn, maybe the tendencies of FFP-related artifactual damage to maybe help separate out artifacts from truth?
Yeah, I think definitely that's, if you have a specific FFP data set and train on that for FFP targets, that should be what it can learn. Yeah, then we can actually did that a little bit internally. Yeah.
Okay. Well, Mohammed, again, thank you for your time and for your presentation. I know I've learned a lot. I'm sure the people who have attended have learned a lot as well, and very excited about our developments in calling somatic variants, which are so very important in cancer and determining the right driver mutations or potential driver mutations so we can improve precision medicine. So thank you for your time.